And one more announcement. So the Nobel Prize for Chemistry was announced yesterday, and it was awarded to Jennifer Doudna and Emmanuel Charpentier for the development of a method for genome editing, which is the CRISPR technology. And I'm going to make sure to highlight CRISPR in a future lecture about biotech and engineering plant resistance to pathogens. So stay tuned for that. And I guess this photo was close to when Dr. Doudna heard that she had won the Nobel. And it's really nice to see that level of excitement, especially this year. Okay, so today the outline of what we're going to be going over in the lecture is we're going to be focusing on evolution and ecology of bacterial plant pathogens. So we're going to start with the general mechanisms of bacterial evolution, and then talk about the lifestyles of plant pathogenic bacteria and transmission routes of plant pathogenic bacteria through a lens of convergent evolution. And this last section might get bumped till Tuesday, depending on timing. But then we're going to talk about the evolutionary trajectory of fastidious obligate pathogenic bacteria, because there's some very interesting ways that their genomes evolve and that their behavior changes as they become, go down this evolutionary trajectory of becoming an obligate pathogen. So evolution is the change in inherited traits or genes in a population of organisms over time. And it can be, so it's not, evolution does not have a goal. It's literally just the change in those inherited traits. The natural populations of organisms have genetic variation amongst them. And we'll talk about the mechanisms of where that genetic variation comes from, but those populations have variation. And then the environment can select certain traits that become more dominant, but there can also be random processes, that occur that change the proportion of those traits in a population. So bottlenecks are not an active selection, but when a pathogen comes into a new region, it will come from an area where that pathogen has a lot of genetic variation, but then only a few individuals generally found that population in the new environment. And so there's a bottleneck of the total genetic variation. There also can just be random chance of genetic drift, where some individuals might be, land in a more nutrient-rich environment and might proliferate more, and other individuals might end up in an environment by chance that leads to them not being able to grow and divide, and so they might be there might be this random chance genetic drift phenomenon that changes the proportion of certain traits in a population. So with bacteria, there's several sources of genetic variation in their populations. The first one is that as all organisms grow and divide, spontaneous mutations can occur. And this can be due to the imperfections of the DNA replication machinery. Bacteria do have protein machinery to repair some of those mutations, but the repair mechanisms aren't perfect. And there can also be recombination of genes and movement across the genome. And recombination methods can end up leading to duplications or loss of certain genes. So recombination can lead to these indels, these insertions and deletions. And a really powerful thing for the generation of new traits and behaviors are when genes get duplicated and then can go down different evolutionary paths via selection. But so if you're looking at genomes of different organisms, you might see that there are point mutations, single nucleotide base pairs that are different. You might see these insertion and deletions of small or large fragments of DNA. And then you might just also see that the genome has become rearranged without any change in the total DNA content. Additionally, with bacteria, they can undergo horizontal gene transfer, which we mentioned in the last lecture. So if you remember, there's transformation, which is the ability for bacteria to take up naked DNA from their environment. And then there's also multiple mobile genetic elements that can facilitate the movement of genes between different bacteria. Plasmids are a very common mobile genetic element, and so are these bacteriophages. So the bacteriophages are the viruses that can infect bacteria. And it's really the plasmids and bacteriophages that are the most common. So bacteriophages can move DNA via transduction, and plasmids can both move DNA via transformation or conjugation processes, depending on the type of plasmid. But you don't need to know this for this class per se, but I do want to mention that transposons, jumping genes, and integrative elements are two other mobile genetic elements that can help DNA move around a genome. So when we're thinking about bacterial plant pathogens and how they evolve, we can think about selection being imposed by agricultural practices. So if we have certain chemical managements that keep the pathogens from growing, that provides a strong selection on the pathogen to evolve mechanisms of resistance or tolerance to those chemical strategies. Additionally, if we have plant cultivars that are genetically are resistant to the pathogen and kill off the pathogen, the resistant plant cultivars pose a selective pressure to the pathogen to overcome that resistance. And then there's also going to be parts of the pathogen's lifestyle beyond the direct agricultural practices. So the environment is going to be a selection. If it's, if there's a lot of, especially as, climate change happens. So pathogens will also, they'll evolve to evolution. Yeah, selection can act through changes in the environment or just the environmental conditions that the organisms have to withstand. And then bottlenecks, you'll see that when new pathogens get introduced into a region, and that knowing that introductions of pathogens into a new region bottlenecks the genetic variation of that population is a way that epidemiologists are able to look at new emerging diseases and come up with hypotheses about whether those diseases evolved from our recent introduction of a pathogen, or were something we just weren't recognizing before. And then genetic drift is just going to be a passive process, but it still can occur. Okay, so I can't really read there's my title slide. So I want to remind you guys about the bacterial genetic architecture and how it really helps bacteria undergo horizontal gene transfer and evolve. The compact genetic structure is very efficient for bacteria to evolve new functions. So because genes tend to be very dense in the DNA and they're often clustered by function, it really facilitates the ability of a multi-gene trait to be transmitted from one bacteria to another through one of these processes, because all of these genes can be transferred at once. So that's one reason why horizontal gene transfer is fairly common in bacteria and can really provide a selective advantage when it occurs in the right conditions. So I want to go through a couple examples of horizontal gene transfer being involved in evolution of bacterial pathogens. So I'm going to start with copper resistance. And so that copper resistance is a trait, and there are genes that can control that trait. And this is relevant because copper products, copper sulfate, copper hydroxide sprays are a common chemical control for foliar or phyllosphere pathogens, including some bacteria that infect the above ground parts. For bacteria that mostly infect bacteria through the soil, chemical controls tend to not be as common because it's just a lot harder to efficiently deliver high amounts of chemicals to the soil without causing significant environmental damage. But foliar sprays, you can spray it more targeted to the plant, and so you don't have to apply as much as you would have to for the soil. Okay, so copper sprays are a management practice. So any organism, pathogen or not, that's on the leaves of these grapevines are going to have to tolerate or resist that copper in order to survive. And so there's going to be a strong selective pressure. So the mode of action of how copper kills organisms is a little bit complex. But the basic gist is it causes oxidative stress. So it through reactions like the Fenton reaction. And if you've heard about how iron is also antimicrobial, copper and iron have similar processes here, where they can react with hydrogen peroxide and then release hydroxide radicals. These free radicals go around the cell and oxidize and damage all of the components. So lipids get oxidized, proteins, DNA. And if you get enough of that damage, then the cells can't survive. So one of the methods that pathogens have for resisting copper, and I guess not just pathogens, bacteria have for resisting copper are through export systems. So one of these is called the COP-ABCD transporter. And you can see the genes drawn here as arrows, where the start codon would be here and the stop codon here. So the gene is encoded right there. You can see it's a multi-gene structure, COP A, B, C, and D. And they're in this compact package and able to be transported on plasmids between different bacteria. And what these proteins do is they encode a multi-protein complex that goes through the inner and outer membrane of gram-negative bacteria. And they facilitate the export of copper ions from the cytoplasm outside the cell. And so this helps keep the concentration of those metal ions low inside the cytoplasm so that there's less oxidative damage to the cells. Another foliar chemical spray that is applied to control some bacterial pathogens is our antibiotics like streptomycin. Generally, antibiotics are not used for many bacterial plant diseases relative to how widely they're used in human medicine. But there are a few cases where the disease is destructive enough to usually expensive tree crops. And they can apply antibiotics at certain times of the year where antibiotics become something that makes sense to apply as part of an integrated management program. And so one of these cases is to control a pathogen called Erwinia amylavra, which causes fire blight disease of apples and other several other orchard tree crops. And so streptomycin is an antibiotic that they can spray at the flowering time of these trees because Erwinia amylovera will actually be transmitted between flowers by pollinators. And so they can spray it at flowering time to keep the populations of this pathogen low in order to keep this disease from spreading through an orchard. So streptomycin is an antibiotic that inhibits bacterial translation. So translation being the process of the ribosome reading the genes on mRNA. And streptomycin binds to the bacterial ribosome. And so that's important because that means that streptomycin doesn't bind to the ribosome of fungi, it doesn't bind to the ribosome of plants or humans. So it's it's a little bit targeted there and inhibits bacterial translation. And so if bacteria can't translate proteins, they can't grow and divide. Oops. Okay, I think I have that in the wrong order. I don't know. This is where I want to do the class participation. Sorry. Okay, so streptomycin inhibits the ribosome. And there's these other processes that bacteria can have to resist antibiotics and chemical controls. We covered some of these on Tuesday. So I want you to respond with Zoom reactions, keep clicking them so that they can show up. And I'll walk you through how we're going to do this active learning exercise. Knowing that some Erwinia are resistant to streptomycin, I want you to vote with what you think the molecular mechanisms are that can confer the streptomycin resistance to streptomycin. And when you sequence their genomes, what you see is that they have mutations in RPSL, which is one of the many genes that encode for ribosomal proteins. Ribosomes are like 20 or more protein genes, and then multiple RNA genes as well. And those mutations are where the streptomycin actually binds to the ribosome, and so they limit the amount of interaction. And then there's also a pathway that's been characterized encoded by this STRA and B, where these enzymes actually break down the streptomycin so that it's not inhibiting translation anymore. And one thing to keep in mind is that this mechanism of breaking down the chemical is like, if you were a bacteria and had complete choice, you should choose this approach because this tends to not affect fitness in other ways. Sometimes these mutations in the ribosome can affect growth rate of bacteria in absence of the streptomycin. So these tend to be a little bit stronger than these RPSL mutations, but these can still be neutral overall for the bacteria. But these just tend to be less harmful for them. So sometimes there's trade-off where the bacteria have to encounter this really terrible antibiotic, and it's okay to have a weaker ribosome as long as you can grow some. But then when the antibiotic is removed, that bacteria might be less fit and might become less common. Okay, so we will do thumbs up for the heart. So you can do clap, everyone has clap, and then the other emoji, any other emoji for STRA. So for this question, I want to ask you which genes, the RPSL ribosome mutations versus STRA-B, might be easier to transmit horizontally on a plasmid, on a phage. Cool, so I'm seeing a few claps, but a lot of hearts and thumbs ups. I agree with and it's really more of a judgment call. I mean, easier is kind of a, a conditional thing. I agree with the STRAB answer. These genes are very common to be transmitted between bacteria. Although this technically can be transmitted horizontally, there's a very good chance that the core genes, if you get a slightly mutated one into a bacterial cell, there's a chance that it has additional mutations that limit its ability to interact with the native machinery in that cell. So core processes are possible to transmit horizontally, but they're less likely to be successful and become something you see in a genome later. But accessory genes are really easy to be transmitted horizontally and become fixed in a population through evolution. Okay, so now we're moving away from chemical controls in terms of bacterial evolution and discussing how bacterial populations can evolve between their pathogenicity on plants. And this one is a really interesting cautionary tale that was relevant very recently, and is still kind of an ongoing controversy in plant pathology. And what's important to think about for this case study is that any individual population of bacteria might have individuals that are along this gradient of maybe they're net neutral for the plant, maybe they're weak pathogens, maybe they're strong pathogens, but there can be some variation here in the population. And certain taxa are, very along the strong to medium. And other groups of organisms can vary from weak mutualists to weak pathogens. And this case of Rhodococcus fascians is a group that as a species can vary. Individuals can be weak pathogens, but other individuals can be weak beneficial bacteria. So they're really interesting. This is a gram-positive. So they're facultative, with facultative meaning that sometimes they have the trait of being a plant pathogen. And whether they're a plant pathogen depends on whether they have this certain virulence plasmid. And individual lineages can gain and lose that virulence plasmid multiple times over the course of their evolution. When strains are falling along the mutualistic side of where they can be in this continuum, they will be strains that produce some growth hormones that can help plants grow a more effective root architecture. That can benefit the plant's ability to take up nutrients from the environment. But when they acquire what we call the virulence plasmid, then they're able to cause what's called bushy top disease. And here they have a few extra genes, and they still produce plant hormones that induce growth, but now they've produced too much of those hormones and in the wrong places and the wrong times. And we end up getting abnormal development of the plants that's bad for the agronomic properties. And so this virulence plasmid has this gene that synthesizes cytokinins, and those are plant hormones that promote growth. And you don't need to know the specific one, but there are multiple, we do want you to know that there are multiple strategies that bacteria can use to manipulate plant growth hormones. And that can be a beneficial strategy for the pathogen. And these other two genes are not super important, but the combination of them in Rhodococcus really helped it switch from being more of a mutualist to a pathogen. And this was important because of an outbreak of bushy top disease in pistachio trees that is still kind of unclear as to what the actual cause was. But there was some kind of sloppy Cook's postulates that was completed. And some scientists found any rhodococcus on the pistachio plants and said, if the plants have rhodococcus, then they're going to develop this disease. And the orchard industry destroyed tons of trees. And then when people went and looked at which rhodococcus were on those trees, they didn't find this virulence plasmid. And so that was, kind of a mistaken potentially a mistaken identity. Is hard to know because those trees are destroyed and gone. But it was definitely a large economic issue. So it's important to know, if you have a pathogen or not, and to also be careful about, about management practices that can cost the industry a lot of money. Okay, so now I want to talk about bacterial taxa and how some of these traits have evolved convergently. So for the rest of this lecture, I'm going to focus a lot on this cartoonish phylogenetic tree that I drew. And so in this tree, the triangle represents strains that are, or taxa that are fairly closely related. And then the dashed line indicates that there's, an amount of difference between these strains, and it's kind of an arbitrary difference. And so in blue, we have a lot of the gram-negative bacteria. Purple, we have some of the gram-positives and the molecules. And when I was trying to think about how to talk about bacterial diseases, this was the way that I thought would be the most effective for helping you understand how bacteria cause disease on plants, and also be able to apply this knowledge to additional taxa that we don't talk about in this class, but that you might encounter in the future, or that might emerge as new pathogens. Because plants are a pretty, lush environment for bacteria. And there is going to be a very persistent evolutionary pressure for bacteria to evolve convergent strategies that allow them to grow to high numbers in plants. And usually it's that high numbers of bacteria that can lead to damage to the plants, which we see as disease. So there's common bacteria that cause leaf spot diseases, and I actually switched this. I'm sorry, I'm not a great plant pathologist sometimes. So Xanthomonas and Pseudomonas are two genera that cause spot and speck, but the speck should be Pseudomonas and the spot should be Xanthomonas. In practice, these diseases look very similar to me, so you can just call them leaf spot diseases in total. How those diseases actually manifest depends a lot on the specific bacterial strain and the specific plant. So unless you're very familiar with what those diseases look like in your location, you're probably not going to be able to see a new one and know if it's a spot or a speck. But the commonalities of these organisms that cause these leaf spot diseases is that they tend to have an epiphytic stage where they will be living on the surface of the plant. And then they have different mechanisms to enter through the stomata, wounds, sometimes the hydathodes, and then grow in the apoplast. And it's only once they grow to high cell numbers in the apoplast that they start to create those symptoms that we see. So the symptoms here are the browning lesions that can sometimes coalesce into larger lesions. And sometimes, like in this case, you'll also have a symptom of chlorosis with yellow expanding around that lesion. And some of those symptoms are caused by secreted toxins that the bacteria produce and that, infiltrate down the leaf in advance of where the bacteria is spreading and help the bacteria change the leaf environment to better suit itself so that it can follow the toxin. Xylem pathogens tend to cause some diseases that look similar, but the main thing about xylem pathogens is they cause a set of symptoms that are due to these bacteria impeding the function of the xylem, unsurprisingly, since they are pathogens of the xylem. But these pathogens tend to grow in the xylem, and the xylem's function for the plants is to transport water from roots to shoots. And that's really essential for an organism. And so these, what happens are these xylem pathogens often, almost all of them, grow to very high cell densities in the xylem vessels, and that clogs the vessels, reduces the sap's ability to flow through them, and so it cuts off the water flow. Occasionally, there's a second mechanism that can occur, where the plant actually recognizes the pathogen and forms some anatomical structures that grow out from the living cells outside the xylem. And they grow these tylose structures that come into the vessel and wall it off. And so you can think of those kind of structures as being immunity. They can protect, keep the pathogen from spreading throughout the plant. But if you have too much of that, it can be a form of autoimmunity and reduce the function of the xylem. And so there's both these tyloses, and plants also can secrete pectin gels that also slow down sap and slow down the pathogen from moving. Okay, so xylem pathogens clog the xylem, and then I think that this might have tripped up some people in the lab from yesterday, because we didn't directly talk about it yet. So if you need to edit your lab exercise after this lecture, you can. So when you have one sign of a lot of xylem bacterial diseases will be if you take a stem of a plant and you cut it here. So up here, there would have been a whole stem of probably a tomato plant. You cut it and you put it into water. What happens is the bacteria that are all in that xylem vessel and all the xylem vessels, you'll actually see them stream out, and there'll be these wispy white things. So one of the videos you saw was bacterial streaming occurring. So in that lab, we ask you to say, now that you're seeing this sign, you're seeing all this bacteria and polysaccharides coming out of the plant, why would you think that would cause wilting symptoms? And so the way that the actual, symptoms of water stress can look different on different plants. So symptoms are all based on the plant physiology and how the plant is responding to or just malfunctioning due to pathogens. Symptoms have to do with plants. And so the really common manifestation will be a wilt. If you have a plant that's herbaceous, you end up, when there's not enough water being transported, the plant will lose the turgidity. It will become not turgid. That's really hard to say. It'll become flaccid. And then, green tomatoes, peppers, they'll droop and wilt. And, you can see healthy plant here and really wilted plant here. But water deprivation symptoms can look different on some other plants. So in some of the grasses and monocots, what you might see some drooping, but you might see a lot more of just these kind of blight scorch symptoms that don't look, like that also could be from lack of water, but they tend to progress. And it tends to follow the venation, the veins of the plant, as that pathogen is moving systemically down the leaf. And then there's also leaf scorches, where you get the drying of the margins of some of these diseases. And eventually in tree diseases, you might see some scorch, but if you weren't there to see this stage, you might just come and see, a dead tree. Because the wood is still there. The wood doesn't droop immediately. but it lost its ability to transport water. And then cankers can also happen if the pathogen both colonizes the xylem and then causes a lot of damage. And then what I've highlighted here are, you can see the diversity of bacteria that have evolved this lifestyle of being a xylem pathogen. And it's happened in multiple clades and even more than this. Soft rot diseases are also a very important bacterial plant disease. So these are diseases of bacteria that invade plants via the soil. And these diseases cause a lot of damage to the starchy tuber organs that we like to eat. So carrots, potatoes. And there are a major group of soft rot bacteria. Most of them are pretty closely related. Other bacteria that you might know of in this broader triangle would be E. coli and Salmonella, but those are not soft rot bacteria. They just have some similar biology. They're second cousins. So those will be the main players. But if you have any given rotten potato, it'd be a little bit hard to know exactly what's causing soft rot, because there are some bacillus. So there are gram-positives and Bacillus and Clostridia, as well as some other gram-negatives that are more closely related to this guy that can cause this kind of symptom. But these are Pectobacterium, Dicea, and Pantoa are kind of the major players in soft rot diseases. And the strategy that these organisms use to cause this disease is really dominated by being pectinase experts. They really these pathogens all produce similar symptoms because some of these genes can be acquired. Okay, so we also have gall and tumor diseases, which can be caused by agrobacterium. And agrobacterium is, so you've noticed I didn't really call out any convergent evolution, and that's because agrobacterium is a really cool bug. Technically, this is actually probably supposed to be multiple genera though. That might change in the future, but we'll mostly call it agrobacteria. Agrobacteria has a really unique strategy for causing those tumors in plants. Agro has this type 4 secretion system, or T4SS. And this is a molecular machine that allows agro, and it actually looks like this. So you've got your inner membrane, the outer membrane of the bacteria, and then this is the membrane of the plant cell. And this molecular machine, many proteins here you can see in all different colors, allows the bacteria to inject into the plant cell. And the cargo that this transports is actually DNA from the bacteria that comes on this tumor-inducing plasmid. This DNA gets injected into the plant cell with some proteins that actually integrate that DNA into the nucleus of the plant cell. So it's a little bit like HIV in terms of the pathogen injecting its own DNA that becomes part of the genome of the host organism. And then what gets expressed off this DNA are plant genes that make the plant produce some of those plant growth hormones, cytokinins and auxins. And it also produces these weird carbon nitrogen compounds called opines that most organisms do not consume, but that agrobacteria can consume. And so it creates, by inducing all of the hormones, it makes a really nice area for the bacteria to grow in the plant, and also makes a lot of food for the bacteria. And the important thing is that even if you were to treat this plant with a ton of antibiotics and kill off all of those agro that were in the tree, or you were to remove some of the symptomatic material, it's kind of like cancer. If you don't remove the whole area, then this DNA is still going to be there. And so even after this pathogen is gone, the plant will still be expressing these genes that lead to those symptoms. And the convergent strategy, so Agrobacterium is the only one that does this type 4 secreted process that genetically modifies the host. But the general strategy of producing plant hormones is a very common one for bacterial pathogens. We've got the bushy top disease that we talked about, the agrobacterium that causes these tumor and galls. And this, whether you call it a disease, is kind of subtle, but it is a symptom that reduces consumer preference for pears. So there's bacteria that can live on the fruit of the pear. And if you have a lot of auxin producers, then that causes the flesh of the pear to become kind of leathery and tough, and consumers don't like that. So pear growers, try to control the amount of auxin producers on their fruit so that they can, consumers will buy more of it. It still tastes fine, but the texture is a little bit less good. Okay. And then there's also phloem pathogens. So one of these is Liberibacter. And we're going to be talking more about Liberibacter in the case study on Tuesday. And we talked about the phytoplasma that cause the poinsettia bushiness. Phloem pathogens are difficult to describe because they change so many traits in plants. So the thing to really remember about phloem pathogens is that they really change a lot of traits in plants. They're complex diseases. And so remember that the phloem is responsible for transporting the sap, the sugars from the photosynthetic parts of plants, the leaves, to the rest of the plant. Another phloem pathogen is this really beautiful phytoplasma that causes phyllodes symptoms. And so this strawberry is actually producing vegetative tissue where it should have seeds. And so it's not sprouting, but it's, yeah, you probably would not want to eat that. It'd probably be fine, it just wouldn't be that tasty of a strawberry. But it's one of the most pretty plant symptoms. Yeah, and then we'll be talking in a lot of detail about Liberibacter, which is causing a very serious disease of orange trees in Florida. And another important thing to remember about phloem pathogens is that they're, one thing that makes them special is that they actually are living inside the phloem cells. And most plant pathogens, and especially most bacterial plant pathogens, are not actually inside the cytoplasm, inside the plant membrane. Most bacteria are in the extracellular spaces, and that's partially because it's easy, for most pathogens, it's easier to move from spot to spot in the plant if you're in those extracellular spaces. But the phloem allows the bacteria to move, they'll end up moving up and down between the pores of the phloem cells. And I said only because biology always has exceptions, so. Okay, so I think I'm going to go into this part because we'll have a little bit of time on the convergent evolution of transmission, but the evolutionary trajectory of the obligate parasites, I'm going to save until Tuesday. Okay, so transmission, when we're thinking about that, we're going to be thinking a little bit more about ecology of the bacteria and how they're able to move between plants. The major modes of transmission are, the water is a very key thing for moving bacteria around plants. So if you've got a lot of bacteria in a canker here, during a rainstorm, the splash and drip damage of that can transmit the bacteria to new parts of the plant. Especially for soil-borne diseases, irrigation and flooding can move. If you have the pathogen contaminating your irrigation water, you can inoculate your whole field. For foliar pathogens, the overhead sprays can be problematic. If you have the pathogen there, that's actually, in the lab, a way that we inoculate plants is with a spray bottle in your plant. So if you have pathogen in your irrigation water, that can really cause issues. Insects will transmit a lot of the vascular organisms, not all of them, but some of the xylem and pretty much all of the phloem pathogens will be transmitted by insects. And then there's occasionally insects that do the contact transmission of pathogens. Some pathogens can invade the seed of the plant as it's developing and be transmitted to the next generation. So for those pathogens, seed companies have to be very careful to make sure that they screen their seed to make sure there is not a high level of pathogen that they're going to be selling to farmers. And then infected transplants, if you have an infected plant, you will move it into your field. That goes for any pathogen. And then for soil-borne bacteria, tractors and plows that pick up dirt and the dirt gets caked onto the machinery, that can move pathogens between fields. And then any sort of instrument that you can use to wound plants can move pathogens between plants. So there are many soil transmitted bacteria. A lot of those soft rot bacteria are transmitted through the soil. Agrobacterium is also good at surviving in the soil and causes a lot of galls in the crown of plants. So that's the portion of the stem that's where the roots meets the stem. So agrobacterium is called crown gall disease, or causes crown gall disease. Ralstonia wilt pathogens are soil transmitted and will invade the roots and then gain access to the xylem, where they'll then grow and spread. And streptomyces are some gram-positives that are soil transmitted. These cause a lot of issues in potato. They make basically just cosmetic lesions on tomatoes called potatoes, sorry. Cosmetic lesions on potatoes in the soil. And so unsurprisingly, a lot of the bacteria that cause damage to roots are good at surviving in the soil. And so if you're thinking about controlling these bugs, focusing on clean irrigation water and sanitizing equipment between fields can be useful for keeping them from spreading between fields. Okay, so there's also a lot of insect transmitted organisms. Erwinia, we will talk about two different species of Erwinia. And my goal there is just to highlight that we don't want you to necessarily know the species level difference, but just that, Erwinia amylavra and this other Erwinia tracheophila, cause different diseases and do have different ecology. So Erwinia amylavra is the one that causes fire blight disease, and that can infect the flowers of the plants. So unsurprisingly, it's able to be transmitted by pollinators that get coated in the nectar, and then can move it to a new flower. And then yesterday I mentioned this Erwinia tracheophila. That is one that is actually transmitted by these herbivorous beetles. And it gets transmitted to the new plants by the beetles defecating the bacteria onto the leaf surface around where they're chewing, and then the bacteria can migrate into the plant. And that one's an actual, highly evolved transmission method, because this Erwinia strain will overwinter in those beetles between seasons. Xylella is a really famous xylem pathogen. It's causing a lot of disease of grapevines in California, Pierce's disease. It's also caused a recent epidemic in olive groves in the Mediterranean that is probably gonna kill off a lot of these ancient, beautiful olive trees where we get all of our fancy olive oils from. And these are transmitted by sap feeding insects that feed on the xylem sap. In California, there's a sharpshooter insect that transmits that. And in Italy, I think it's something called a spittlebug. And the specific identities of those insects are not important for you to know for this class, but know that they are insects that feed on xylem sap, because they'll be injecting the bacteria into the xylem vessels. Liberobacter is a phloem pathogen. Unsurprisingly, the insects that transmit them are insects that feed on phloem sap. And I'm currently blanking on the name of those insects, but we'll be reading about them in depth for the case study. This one I've made small text because it's like really not common to be transmitted by insects, and so you probably shouldn't remember this. But as someone who is a Ralstonia expert, I couldn't resist putting it on the slide because there's a few rare cases where pollinators can transmit this usually soil-borne bacteria. And then Rhaetiobacter is a really cool one that is causing a lot of problems in Australia right now. And the reason I think it's cool is it's a nematode transmitted bacteria. This Rhaetiobacter produces some toxins in grain seed. And these seed gall nematodes will move it between wheat plants, some of the different cereal grain plants. So the nematodes actually will come up from the soil and go all the way up the plant and then deposit the bacteria in the seed. The bacteria makes a toxin, and this toxin causes paralysis and death of sheep and humans if we were to eat it. So that's problematic. And then phytoplasmas are also phloem pathogens that are transmitted by sap feeding insects. And so these two phloem pathogens of phytoplasma and Liberobacter, although they're very unrelated, we've got one that's a gram-negative and one that's more closely related to gram-positives. They end up causing similarly complex diseases with similar symptoms because they are causing disease of the same organ in the phloem. And then we have a few seed transmitters. So these bacteria are ones that are known to be able to infect seeds of plants. We've got Xanthomonas, Pseudomonas, Clavibacter, and you don't need to remember Acidovorax, but I felt like I had to name drop it because it's a very important one for melon seed transmission. So if you ever have, a melon blight, you'll be running into that guy. And then all pathogens can be transmitted in live plant tissue. And that's it's a very obvious thing, but it's also an important thing to think about as far as the transmission of these bacteria between fields. And, when you come into California, California's pretty serious about keeping our agriculture. So the key points of this slide are that a few agriculture systems like potatoes and banana production are particularly at risk for pathogen transmission because vegetative parts of the plant are used for propagation. So these are not plants that are propagated by true seeds. True seeds are actually very effective at limiting most pathogens from invading them. But the potato tubers are cut into pieces that act as seeds that are planted for the next crop. And in bananas, these sucker trees will grow out of the base of the plant, and then you can cut those off and propagate them into the soil. So these create problems for just potential risks for moving pathogens during production. And then another important thing globally is the ornamental plant trade. So a lot of the ornamental plants are plants that are propagated by cuttings. And a lot of them don't produce seed with very high efficiency. So geraniums and a lot of other plants are often grown propagatively in warmer parts of the world that have longer days. And then get moved all around the world for people's gardens. So that's a potential risk for moving plants, moving pathogens into new areas.